Assalamu alaikum dear students. Today our topic is another growth hormone, that is abscisic acid. We will study about its introduction, discovery and how it is transported and why it occurs. As far as introduction is concerned, this abscisic acid it is a plant growth hormone. It comes in the category of plant growth inhibitors. It functions in many plant developmental processes, including seed and bud dormancy. It controls the organ size and also causes the cytometal closure. Actually, we see this hormone or this growth inhibitor, this is important for plants because it prepares the plant for the responses to environmental stresses, various environmental stresses, including the conditions during the harsh winter, including drought, soil salinity, cold tolerance, freezing tolerance, heat stress and heavy metal ion tolerance. So this hormone, it is important, otherwise the plant will die. So this hormone or abscisic acid, this prepares the plant to survive during the harsh conditions by performing different roles or by showing different physiological conditions. Then as far as its discovery is concerned, this hormone was also discovered while the scientists while working on different these plants, or they see the effect of this substance on different plants. For example, in 1940, scientists isolated a substance from sycamore leaves and they called this substance as dormins. Hamburg was the scientist. Then in 1960s, Eagles and Philip Waring confirmed that application of dormin, this substance or dormin, to a bud, it induces the dormancy. Likewise, Adicott in 1963 discovered a substance stimulated abscission of cotton fruit. He see the abscission of cotton fruit takes place and he found there a substance and he named this substance as abscissin second. In 1964, it became evident that the three groups had discovered the same plant hormone, same substance, that means Hamburg, Waring or the Adicott. Later on, this substance or the name was changed to abscisic acid. So this is a single hormone, that is the abscisic acid. This is about how it was discovered. As far as its chemical composition is concerned, abscisic acid is a 15-carbon sesquiterpene compound. It is composed of 3 isoprene residues, cyclohexene ring with ketone or ketone 1 hydroxyl group as side chain with a terminal carboxylic acid group. So it is a sesquiterpene. We see the orientation of carboxylic group at carbon 2 determines the cis and trans isomer of abscisic acid. As far as cis abscisic acid is concerned, it is biologically active than trans abscisic acid. Nearly we see all the naturally occurring abscisic acid, it is in the cis form. We see here this is the naturally occurring cis form of carboxylic acid and this is the trans form. Cis form is active form. Now we will study how why it is occurrence and how it is distributed in plant tissues. We see this abscisic acid, it is a ubiquitous plant hormone that is present in all vascular plants. Even we see in bryophytes, it is found in mosses but not in liverworts. Some fungi, they synthesize this as secondary metabolite. In algae or in liverworts, compound 15-carbon compound that is known as linolenic acid, it has been found just like it has structured just like ABA. Now, where it is synthesized, ABA or abscisic acid, it is synthesized in all types of cells that contain chloroplasts or other plastids. That means chloroplast, where it is present in all plant cells, abscisic acid is synthesized. It occurs predominantly in mature green leaves, and we see it has been detected in major organs or living tissues from root caps to apical bud. Even in phloem or xylem, it is also present. So we see its occurrence and distribution. That means all vascular plants, even in mosses, even as secondary metabolite as fungi, then in cells where chloroplasts are present, and predominantly in mature green leaves. So hope students you understand this. Now, how ABA or abscisic is transport in plants. We see externally applied ABA when it was externally applied to plant tissue or plant, it distributed in all directions. That is, cell to cell transport is slow. ABA synthesized root cap transported to central vascular tissue. We see this transfer mostly occurs in the free form. That means not with any conjugated group. If it is conjugated form, it mostly as ABA beta D glycosyl ester. Then the pH gradient also influences its distribution, or we say how pH gradient influences its dissociated or undissociated form. We see at low pH protonated or undissociated form of ABH present, whereas at high pH dissociated form of ABA is present. So as far as transport is concerned, it distributed in all directions, from root cap transported to central vascular tissue, mostly transported in free form. Hope students you understand all this about the